Thanks so much for coming to my presentation. My name's Cameron Skinner, and I'm a product manager here at Millennium Science. And I work with the Agilent BioTek technology, which is front and center the Lionheart instrument, which we just installed up at CMCA. So this presentation will go through the Lionheart itself and a few other things. So just to start out, just acknowledgement of country. Millennium Science acknowledges the traditional owners of this land, which is the Noongar people. We recognize them as the first innovators, scientists, and storytellers, and we pay our respects to their elders past, present, and emerging, and extend our respects to all Aboriginal and Torres Strait Islander peoples. Just going to hide this toolbar. Hide floating. All right. So here today is Louise Eastness, who's in the back, who is the territory manager for your state, South Australia, and also Northern Territory, and is based in South Australia. And I'm based over in Melbourne, which is, I think, quite dreary at the moment. So glad to be here in sunny Perth. I deal with Agilent BioTek, so a whole bunch of things which I'll go through, as well as Lionheart Bio, as well as a gene editing company known as Horizon, which is a Dharmacom brand as well. So again, the overview will go through the Agilent technologies and a bit on Millennium Science as a whole, understanding Lionheart imaging systems, the features and functionality, as well as showcasing some of the diverse applications that can go on this instrument. Flash on the screen is one of the initiatives that Millennium Science has developed, and we can basically assist as a distributor company. We can assist with sample prep all the way through to translational research. So I've showed this to a few other researchers. So any feedback that you have in terms of what you see here is would be amazing, since this is still in development. But essentially, you'll see just down below, Agilent spans the entire workflow that you can see down here. And another initiative that we like to call Blissful, which includes some of our named partners, which is BioLine Meridian Bioscience, long read sequencing through PacBio, imaging, which is front and center through Agilent, spatial with our 10x partner, as well as flow cytometry with also Agilent. Taking you through just some of the stuff that I do. So the automated cell imager is exactly what the Lionheart does. And we also have microplate readers, automated liquid dispensing and handling, as well as microplate automation. The other part of Agilent that we deal with is also the Acceligence and Novocyte flow cytometry instruments. So the Acceligence is a really cool instrument for label-free cell detection, as well as the Novocyte flow cytometry, which is also traditional flow, as well as their new spectral instrument called the Opteon. Launching straight into the Lionheart instrument and its core features, it's a very accessible and versatile platform, which I'll show with some of the applications. It has an automated 6-objective turret, and I'll go through the objectives that are on the system. The objectives that are in the system are on another slide, but simply they range from 1.25x all the way through to 60. And there's also an upgrade ability to purchase a 60x and 100x oil immersion. It's an open stage, so you're able to throw in a whole bunch of different labware and different periphery attachments. And there's several adapters that can actually go on the instrument as well. It's also T75 cell culture flask compatible, and it's all powered through the Gen 5 microplate reader and imager software. Inside the instrument, going through around this in clockwise, the transmitted light optics right at the front with the stage carrier. The 6-position objective turret lives in a section just down below. And then we also have the filter cubes down here, which there's 4 filter cubes installed here, as well as the camera, which sits at the back left-hand corner. And what's missing just from this is a cover for any live cell experiment, just to keep them nice and happy. The imaging modalities is fluorescence, bright field, high contrast, bright field, phase contrast for the phase objectives, and color bright field. So the magnification range, as I mentioned before, as they flash across the screen, it's just a nice picture of the same cells with different fields of view. So the ones highlighted here in orange are the ones that we've installed on the instrument right now. Yeah, I think it's a really, really cool animation to just go through. And the wide field imaging filter cubes that are also installed on the instrument are DAPI, GFP, TRITSI, and Sci5. So I've just highlighted them along here, and I'll provide you with the slide so you have the excitation emission. But you'll see here that I've left the part numbers for the good reason that a lot of our filter cubes can go on the existing LEDs. So you don't need to buy the whole package because they come in two different parts pictured down below. So you're able to sort of have a bit more modularity around the instrument. For bright field imaging, high resolution, as well as ideal for cell culture assays, you can see here that we have an open aperture, passes through the condenser, through and hits the camera, and then you're able to resolve the bright field image, which is as simple as it gets for bright field imaging. For fluorescence, much of the same way, where we've got an automated track for the different filter cubes. You can see here that's flashing on the screen, LED fires, it moves through the mirrors and hits the sample, and we can actually resolve that image and overlay those colors all together. So you're able to do 4 colors in the experiment with the fluorescent channels. Color bright field, much the same way, where we have RGB LED image capture, so ideal for histology applications. So here we can actually see, the image be resolved quite quickly, much in the same way that bright field. On the system, there's what's called image-based autofocus, and this is a really flexible option for a whole bunch of different biology. So here we have the fixed and the live biology here, and you can see where the peak is, where we've got sort of the maximum peak of that fluorescence, where we can actually go and resolve that sample with those 3 colors, and then it can move on to the separate well, which might have a different sample altogether. So again, recommended for 3D cell culture, thick biology, kinetic studies, moving biology, and fixed samples as well. So the labware versatility, there's many, many different types of adapters that can be placed on the instrument. So the ones that are on the instrument currently is the standard stage. And with that, there's also a 2-slide holder that is at the instrument. The humidity stage is also available on the instrument, as well as the multi-vessel adapter for anything longer than a slide or anything shorter than a slide. Part of the instrument as well, when it comes to live cell, again, keeping them nice and happy, is temperature and CO2. So the temperature and CO2 on the left-hand side, you can see it's simply just a flick where we turn on the instrument and we can actually modulate that CO2 and O2 going in. And CO2 will be installed and up and running for your assays. Since we can install that now with gas, O2 will be through LISA if we have any of those projects going on. But yeah, the O2 and CO2 controls are just down below with 0 and 20% and 1 to 19% if you need those assays run. The temperature control, where we have up to 45 degrees as well. In the middle, which I'll show you on the next slide, which is just some oil immersion examples, are only on the 60x and the 100x objectives. So really looking at intracellular features. And the last animation on the right-hand side is something that's another upgrade, which is called the dual reagent injector module. Simply put, you can put reagents on the side that can automate injection of liquids or reagents from 5 to 1,000 microliters, and they can either be angled or offset tips. So depending on what labware you're putting inside the instrument. I love, since it's a microscopy talk, you can't not show beautiful images. So here, some endosome staining and some focal adhesion staining as well. For the oil immersion, it's just improved resolution that you're only going to get from the instrument, as well as some of the 100x oil objective images where we have the mouse cortex as well as the mouse cortex on the other side as well. So as part of the Lionheart and also part of the Agilent story, it's all about building something to publish, I suppose. So whatever you're placing on the instrument, it can either be part of a drug screen or any sort of screen that you have, the 2D live cell behavior, a 3D model, fixed cell markers, cell analysis, or even some histology confirmation. All of these are a road to publish that we can actually perform inside the instrument. And I just wanted to flash up just part of the internal software. This is the Gen 5 workflow. So as soon as you arrive to the instrument and you start the software, you can simply click using an existing protocol, or you can design your own protocol, and away you go. So here in this example, we're using an existing protocol, and let's just say we wanted to count cells with two colors, in this case, DAPI and GFP. So pictured here is a citation, but it's much the same for all of our imaging technology. So when we load the plate onto the instrument, we can then acquire the images as per the channels you require. And then you can actually have an analysis that's either ongoing or can be performed at the end. And this is where you can obviously receive those metrics as soon as the instrument has finished its imaging cycle, or as soon as you wanted to export them. So the power here is not only imaging on the fly, but also analysis on the fly to make sure the cells haven't died accidentally, or if anything sort of issues with your experiment. So it's all through Excel, which simply you can place into your pipeline, such as R or any of your other pipelines. Much like the workflow slide at the start, there's a lot going on with this slide when it comes to Gen 5. But as part of the capture portion, essentially when we can capture, we can process onboard the Gen 5 software, and that could be background flattening, a deconvolution, adjusting the brightness and contrast, as well as a digital phase contrast. We can analyze using a primary mask and a secondary mask and look at object level objects, or object level for just say cells, for instance. And here is where we can also look at label free, spot counting, a whole bunch of different assays. So as part of the capturing, we can just capture single. We can also capture multicolor. We can montage on the instrument. We can also perform Z stacking. And in conjunction with all of these, you can build a nice comprehensive story, especially when we're performing kinetics and over time life cell experiments. The last thing as well is it's not locked with any proprietary software. So if you wanted to export and perform this analysis in Fiji, or if you want to perform it on another platform, you're able to easily export it in a TIFF format. I'm just going to launch into a few applications I just cherry picked from a few of the Agilent sort of repertoire. There are about, I think, 200 applications. So I just wanted to pick out maybe seven or eight to go through. But as you can see here, there are a whole bunch of different applications that can apply to the Lionheart instrument. When it comes to a screening application, I know a lot of work has been going on with 3D bioprinting, especially when it comes to cell migration assays. So here is where spheroids have been plated out in a 384 well plate, and they've been imaged using a 2.5 objective over 48 hours at sort of a regular interval. So all of these are captured and processed within Gen 5, and then you can actually use the onboard masks to interpret some of the results that you can have. So in terms of the screening, if you're attacking them with any drugs, or if you're attacking them with any cell types, you're able to export the data immediately and generate graphs within the software. As part of the T cell mediated cytotoxicity, you can see a few animations up here, but Z stacking is really important when it comes to 3D, or if you're looking at large biology. So every 4 hours that we were tracking the T cells and looking at a tumoroid sample here. So cell interactions and labeling with an apoptotic marker, as well as a necrotic marker for cell induction. And here is where movies can be made from the Z-stacked images, and the kinetic can actually be sort of overlaid, allowing for the visualization of those cells actually killing the organoid. And you can actually, again, process them and publish them at the end. So when it comes to anything that isn't able to be labeled, we are able to utilize either the digital phase or the bright field modalities. And again, looking at the spheroids, again, a bit of a trend at the start where I picked 3D. We can capture, and not only capture in Z, but also montage the sample itself. So they can be stitched automatically. They can be processed in terms of the Z plane, as well as areas covered by the total invading tumoroid. We are able to look at that and the propagating cells calculated at the bottom. My simplest application note, and I think my favorite application note, is just literally measuring apoptosis in zebrafish treated with ethanol. I mean, you would think, that sounds easy, but again, zebrafish, much like other organisms, they're not in the center of the wells, and we're able to actually track them over time on different plates and actually place beacons across the plates. And that just means that if it's in the corner of one plate or in the corner of one well or in the middle of another well, we're able to track them. So here we can image them over time using a 2x and Z stack over time and analyze them as simple as it gets, like this. Quantifying morphology is very important when it comes to this type of study, especially if it's in conjunction with toxins. So anything like circular, curved, short, or straight, Gen 5 is actually able to not only process and montage using different objectives, but able to look at the shape and actually distinguish what those shapes are and count the shapes and quantify them quite quickly. So when we actually place this type of experiment onto the instrument, you can work with, just say, Louise or myself to help you get started with the analysis. And simply when you return to the instrument, you're able to have that workflow already processed for you and all the analysis is already set up for you. Angiogenesis is much the same, where in fluorescence, we're able to look at different parts of an organism, such as the zebrafish. So here we're using a 10x objective, and this is a bit longer, where every 45 minutes, we're sort of taking an image for 24 hours. Images are processed in focus or and also Z stacking, and a background subtraction has actually been applied. So this is only to enable the primary mask accurately sort of thresholding around the area of the fluorescence down below, and you're able to track the vascular area in this case within Gen 5 for in the context of some inhibitors. Wound healing for cells is applicable, as well as wound healing on the zebrafish as well. So here is where some zebrafish have been injured, and then we are able to track how well it's being healed over time on the instrument. So anything high throughput, and we're thinking like in a plate context, we're able to look at, especially when it comes to migration assays. So if we wanted to simply look at the detection zone within the middle, and we wanted to see how many cells migrate within the center in the context of drugs or in the context of different cell types, we're able to publish that, and we're actually able to perform what's called a plug. So the image at the top and image on the second row, that's looking at the global image, but a plug can be used inside Gen 5 to only look at the center of where you've actually performed this wound heal, or this sort of plug essentially, and then you're able to see where that cell-free zone over time accumulates cells, and then we're able to quantify that. For something that you're unable to utilize with cells without any labels, there is a technique called label-free confluence using high contrast bright field, and it uses two stages where it images a sample at the level of focus, and then it will reach a sample at the top. So it basically illuminates the cells in a way, and we're actually able to threshold them a little bit better if we weren't able to do that. So this basically just means that you don't need to add on any thresholding or add on any fluorescence that might kill your cells. Something to look at, which is also something which is really cool, phagocytosis assays. I think for if you're, again, in the screening context, or if it's only one well context, you're able to perform not only one channel, but two channels. And essentially, if the kit allows for the fluorescence channels, we can basically perform any of the imaging that you require. So the movie here, which is just playing right there, is the kinetic movie showing an increase of the bioparticles uptaken by the cells. We can actually look at that and plot that over time. Another one of my favorite techniques as well is automated transfection efficiency. So this is calculation based on microplate. So increase viral titer over the plate. We're actually able to look at the cells, not only look at the cells, but background subtract, and obviously look at the green cells and the green transfected cells. So you can see here, as it obviously moves through the plate, we're obviously getting more transfection. I wanted to end with the applications here, and I wanted to just sort of give you a bit of a flavor of what Gen 5 looks like, since I've spoken a whole bunch about the analysis software. Was there any questions from the audience here before I launch in on any of the applications? No? Easy. Okay. Into Gen 5. As soon as you launch into Gen 5, simply the task manager appears. The task manager essentially runs the instrument where we can move in image and manual mode, where we can simply capture now. And that just means that we can move along the plates as we would a normal microscope. So looking down the binoculars, going to C4, screening around the plate to see what's actually happening or if the fluorescence has worked in a way. Where I think a lot of researchers will be living is the experiments portion. And this is where the high throughput nature comes into it. We're able to create an experiment with your particular protocol intricacies, and we're actually able to look at different protocols. So here, if we create new, and we can just create a sample protocol, the protocol is basically a tree. So where we have sort of a tree format. So basically from the top, it's procedure, plate layout, data reduction, and then export. As simple as that. And where procedure is, we can simply select the Lionheart instrument. And here is where everything is open. The steps and the actions on the left-hand side are simply you can image, set temperature, dispense, and gas recharge. The gas recharge we'll talk about at the instrument itself, but simply the first thing that we do is basically select the labware. And there are many different types of labware that can go on the instrument, and we can actually perform, if you've got something bespoke or if you've got something 3D printed or something very, very special, we're actually able to perform a custom labware if you need. So simply we can select 96-volt plate, and for any of the instruments, let's just say we want to run maybe like a kinetic assay, or we want to start with a temperature, we simply click set temperature. And here is where we set it at the desired temperature. So here is where it's checked preheat before continuing to the next step. That just means that before, or it needs to reach a certain temperature before it starts imaging, for instance. And we can click OK, and you can see the temperature set is nicely labeled. If we wanted to start imaging, we can simply just click OK. And this is where the imaging steps start. We can select the objective that we would like. And here is all the objectives that you can see. And again, just a reminder, it's 4, 10, 20, and 40x. So I can just simply click the 4x, or if I wanted to select the 4x, the field of view down the bottom right-hand corner changes. When it comes to binning, autofocus binning, and capture binning, I think you'll lean on the microscopy staff if you embark on sort of utilizing that. I say don't really use those settings, even if the fluorescence is quite dim. I just feel like it's the easiest thing because we don't want to actually minimize the data that we're actually receiving. If we've got a few fluorophores on the instrument, we can simply click the fluorophore utility. And here is where we can select the fluorophore. And a whole bunch of fluorophores are already populated, and we're actually able to insert fluorophores from some of the configurations. If we wanted just to select two colors, we can select Alexifluor 405, and then we can actually select the color that we want to actually look at. So here is where you can see that we're appropriately imaging that fluorophore based on the excitation and emission. And here is where we can click plus, and this is where we can start adding on different fluorophores. Just a representation, or sort of just a note here. Blue, red, green, and let's just say a purple color. It doesn't mean that that's going to be the color on the image. It's just a pseudo color for when you have a look at the, basically when you have a look at the peaks, you're able to distinguish between each of them. So if I select, just say this color like this, you're able to simply just populate the colors that you want. And I think it's a really good representation if you've got any overlap between your colors. If you've got overlap between your colors, you might have a bit of spectral overlap and you might not be quantifying what you need properly. So if we've got this situation and we've got two colors, we can simply click OK. Okay, and you can see here the fluorophore utility is populated like that. If we wanted to utilize a bright field or a color bright field, this is where we can select them in the same way. Okay. Auto exposure we can utilize, or we could go through and optimize using this button here. If I click this, it will take me to the instrument portion, and since I'm not connected, it might throw up some issues. So we won't do that. So the illumination is the power of basically the LED that's coming from the system itself, and then the integration time is how long, obviously. And then the gain function is the digital amplification of that signal. So if you're worried about photo bleaching or anything like that, just please talk to microscopy people. When it comes to optimizing bright field, you're able to optimize either with an auto, or you can screen through the plate and just see what your experiment's doing. And this is where you can then start developing quite a complex experiment from the channels. So if we wanted to reduce the plate, and let's just say we only had these, we can simply just click OK, and then it's only going to image those wells. When it comes to defining a beacon, if we define a beacon, we would need to go through the different plate or the different areas and basically select where our biology is. The alternative to that is if we wanted to Z-stack and montage, we are able to simply add in those parameters just down below. And you can say how large can the montage go? We can go 10 by 10. You can get quite a large data set if you don't want to actually go through and put beacons on your instrument. Again, with the Z-stacking, just to make it very simple, it's a wide field instrument, so we have parallel beam of light coming down. We can Z-stack either in the focal plane right in the middle, or we can start right in the bottom and move up, depending on what your biology looks like. And if we just wanted to just simply click OK, that's where we can simply leave the imaging step, so it's all nicely summarized in one line. If we wanted to perform this over time, we can simply click Start Kinetic, and this is where we can select the particular runtime. We can select an hour, and then if we wanted to have imaging performed every 10 minutes, this is where we can select. So the runtime is the total time in hours, minutes, and seconds, and then we have the interval, which is the minimum interval of the imaging. Obviously, if you're imaging 4 colors, a whole entire plate, Z-stacking 5 times, and you're montaging 10 by 10, that might not be the most appropriate interval. You just might need to optimize how often you're imaging, and you'll also receive a whole bunch of data on the computer. So yeah, once we click that, and if we're happy with the interval that's been selected, here is where we can actually add the imaging step within that kinetic step. So we just simply dragged it down onto there, and then we're enabling that imaging over time. When it comes to other options, so plates, plate options, if you wanted to utilize, or if you wanted to add a drug, for instance, or if you wanted to add another cell type on top, we can actually perform delays. So we can actually look at a delay if we set the temperature and I don't want to start imaging till 12 hours. We can simply click delay for 12 hours. So once it's reached temperature, we want to delay for 12 hours, and then the kinetic interval will start. If we wanted to perform or stop the assay, we are able to stop the assay, and you can pull the plate out, and then you can add whatever you required, and then simply away you go. So it doesn't mean that once it's on the instrument, you can't touch anything. Simply, you can perform whatever you need. You can also process anything within well mode or plate mode. So simply put, if you've got the dual reagent injectors and you want to analyze injection of, just say, a reagent, and you need to image immediately or image immediately over time, that's where we have well mode that comes into it. So it's almost like inject, image A1 over time, and then move to B1 or A2, inject, image, etc. Because if you inject over the whole entire plate, you might miss something in that way. We can simply click OK. And then from the plate layout, this is where we can actually start designing what's actually in our plate. So it's actually selected default samples down below. And if we click OK, this is where we can start entering in what sample actually might be in there. It's not necessary to do this. So if you actually don't want to do this, but simply put, it looks really cool in the report when you actually export it because everything's actually collated for you. So once we do that, you can simply put samples 1 through, let's just say 45, either going this way, or we can actually swap the orientation for it to go the other way, just whatever you need. If we click OK, and the data reduction tab, and once we're at the instrument, it will make a little bit more sense because having data to then perform data reduction makes more sense. But here is where you can perform image pre-processing, deconvolution, digital phase contrast, as well as Z projection. If there's any montaging, then it would actually appear here with the stitching step. So all of this can be automated as soon as you place your plate on. Click OK. And then the report and export builders from the analysis that you performed. If you wanted cell count, if you've got subpopulation analysis, let's just say there are some that are blue, there are some that are green, and then some that co-localize. We're able to segment those co-localized cells, and we can actually export them in whatever fashion that you want. So simply put, it's all done through Gen 5 to export into Excel, like I mentioned before. And you simply just follow the steps where you want the system description, the plate description, what you've used. The raw data is very important, the calculated data that you've utilized, as well as sort of the plate format or the format that it's exported. And you can simply click OK, and that's the exporting done. If you've got multiple plates that you want to actually perform on the instrument, you're able to not only just look at one plate, but you're able to add a plate. This just requires you to put the plate onto the instrument since it's only caters for one at a time. Simply put, that's all I really wanted to sort of talk about when it comes to the applications. I'll flash that back up on the screen for the wide variety of things that can be performed on the instrument. As you can see, the examples sort of speak for themselves as to what can actually be run. I didn't go into any cell migration as well. My cell migration assays, when I scratched them, were getting a lot better when I was actually doing it manually. But we've got instruments that can automate this. We've got instruments that can help with media exchange. We've got instruments that can perform a whole bunch of different assays, especially so you're not here on the weekend doing science. So that's all I have for you guys. And I welcome any questions that you might have for me. Yeah.
Migration, you said there's a tool. Is there a like specific tool that does it?
Yeah, it's an instrument that basically has like a little sort of like a needle and just basically does it for you. Yeah. Yeah. Persevere. It'll be fine. Sorry, pardon. Sorry, can't hear you. This one. Any of the application notes, I can actually find more information on the assay. So on this one, I'm pretty sure they only make like a puncture with something. So there are other tools that can just make sort of an area in the cells by like wound healing it. And simply put, this image is the whole global image, and you're able to perform a plug within Gen 5. So it's able to just sort of attenuate basically down to like a disc shape, or even a square, or even any sort of shape that you would like to draw. So you're able to perform that.
The whole scratch.
Yep, absolutely. Yeah. And it doesn't necessarily need to have fluorescence either. You can do it with just bright field. Any questions online? Or oh, sure, no worries. Oh, the network's not very good. Let's have a look. Why not? DAPI is not, the DAPI stain is not required for focusing. So you're able to focus on any particular channel that you require. So you can focus on color bright field, you can focus on conventional bright field, or another fluorescent channel. Simply put, the first channel that I utilized from Gen 5 There we go. So if I go back to procedure, back to image, you can utilize this channel based on the focus settings. This is getting a bit more in depth and it makes more sense at the instrument when we're playing with the sample, but you're able to autofocus and then simply utilize that focal plane for all the samples, or you could use a range of focus depending on your labware, or you could utilize yeah, a different channel if you wanted to. I just simply put DAPI because it makes sense in my brain to start with DAPI. Lisa? I think we were speaking about the O2 capabilities and it needs a cylinder which can be organized through CMCA.
So after the instrument is capable of it, so if
Yeah.
there's the O2 cylinder, we can plug it into the
Yeah.
instrument.
Yeah, it'd be nitrogen. Yeah, it'd be a nitrogen gas. Yeah, that goes in. It's just labeled O2 on the instrument for like the different parts of the gas. Yeah.
Then,
Mhm.
we specialize like for any practice we do.
Any plate.
And
Yeah, any plate. So as long as it can be resolved, like as long as yeah, any labware basically can go on. So as I mentioned before on sort of the Gen 5 area, you're able to select a whole bunch of different labware.
would that tool you just mentioned.
Yep.
So it work with any kind of like
No, there's the automated one works with specific sort of a specific apparatus. And because it has to obviously have a different Z, yeah, with them. Yeah. We can have a chat about what's compatible. Yeah.
on the topic of labware,
Mhm.
for example, using two settings, you have to use specific types of those that are with the adapters we have, and do you know which ones you have?
That I'll have to ask. Yeah, that. So if you have a catalog number or like a specific brand that you like, please email Louise or I, or even through Lisa, and we can organize what that looks like. Yeah, they do. They do.
We know. Us, so,
Yeah. Who do you use?
uh, right.
Mhm. My test plate, my test plate, I think is 6 years old, is a granite plate and it still has color. It still has fluorescence and it's been around everywhere. Yeah. Yep. Yeah. So there's an adjustable one down there that we could fit in. Sometimes it's really good for a biddy because for the slide holders, it's literally built for the standard slide. Some of them fall right through. Or if you've even snapped a slide, the multi-vessel adapter is actually really cool at doing that. Yeah. Yes. Yeah, so the standard stage basically we basically put the humidity on top, or we put like an apparatus on top to make sure that everything stays nice for the cells. And simply put, you just need to come back to the instrument, I think every 2 to 3 days, just to top up the water. Yeah, just on the periphery to make sure it's all nice. Yeah, yeah. You just use a pipette and it just fills around the trough and stuff. Yeah, it's pretty simple.
This one is inverted.
It is.
Also, you mentioned about the old. It's underneath. Is that right?
So the objective, and then the oil will go on top, and then the sample will come together. You would put the oil on, the stage is open. So you're able to drop a bit of oil onto the objective and then place the sample on. Yeah. No all objectives at the moment. No. Yeah. Something to explore, maybe. I hear, yeah. Well, being the 60x and the 100x, they look very particular based on the other objectives. So it shouldn't happen, but I know it does happen sometimes. Yeah, it should be fun. And they're very simple to clean as well. So if you simply just take them out of the turret, use like a stereo microscope, some ethanol, and then you put it back. Yeah.
Yeah, have that one.
Yeah.
Yeah, we can. Things like that happening, and if someone does do that, we do not responsible for that.
So the other question is Z stacking range for each well determined individually. So we actually have it's like a nominal focus that's across the plate. So it would just be if your cell's a little bit higher or a little bit lower. We set the nominal focus before we start the experiment. And as we move into the experiment, where we set the Z is basically at the focal point. So where it's in focus, it will then perform the Z. Because some labware is quite varied between wells, or it can vary quite rapidly between wells. And obviously we don't want that same, maybe like 20 or 30 Zs just to get everything in focus. So it focuses first and then applies the Z stacking. Absolutely. Yeah. Yeah. It's the same as the 3D printing. They sometimes print a little portion first and then the cells exist a little bit higher. And then sometimes even with like the 3D structures, organoids migrate through. Like, yeah. So that can change over time. Exactly. Yeah. And that's the power sort of the imager manual mode. You're still able to drive around the microscope as if it was like a benchtop, sort of thing that you're able to move in with the stage. Yeah. Any other questions? Excited? Yes, Lisa.
How many places is it all places for filters? So if we wanted to have like extra filter, can we, is it something that we could change?
Yep.
For example, if we wanted to do CFP, YFP, can we quite easily change the filters for that experiment?
Absolutely. Yeah. So if you want to CFP, this list is pretty comprehensive in terms of the fluorescence that can go on. And yeah, having the 4 colors, I think they selected the most common. I think that you would see. But if you need anything else, we're able to help you, with the filters. If you wanted to, I think we've got a Texas red filter cube. We're able to loan as well if you needed something like that as well. The deepest it goes is size 7. So the emission is it's a pretty wide emission on that filter cube, which is around 850. Yeah. Yeah, you can utilize what the IR dye. Yeah. Anything else? Well, that's all. Thanks so much for coming to seeing my presentation. Louise and I are here for today and tomorrow as well, so we'll be at the instrument, obviously organized between the facility and get them trained up. And yeah, we'll put our business cards there. Part of the installation, it isn't all going to go through, I think, the facility. I'm also available to help remotely and also perform analysis remotely. So a part of Gen 5, you can export and actually send to me. Not large, like a full 96-fold plate Z stacking, like select maybe some representative images, but even if we're available on like Zoom, like it could just be thresholding's an issue and we just troubleshoot online. Yeah. Thanks so much for attending. I really appreciate it. Thank you.